Phillips, and today we'll be looking at in vivo cloning, continuing looking at our recombinant DNA project. So, in vivo cloning means inserting the gene of DNA into a living organism, and we focus at A level into bacteria. And this is different from making copies using a test tube with in vitro or PCR, which we'll talk about in a future method. We've talked about recombinant DNA and how it only works because making genetic code is universal, so it codes for the same codons, codes for the same amino acids in all living things, and transcription and translation is essentially the same in all living organisms. There are 5 stages of making proteins. Last video we looked at isolation. This video we're looking at insertion and transformation. So how do we get the DNA into the vector? How do we then make the vector go into the host cell? And then we have to look at how we can identify to check the host cell has actually taken the vector. And for this, we could do the 3 methods we talked about in video 1 on this section. To get the gene, we then need to insert the gene into the plasmid and insert the plasmid into a bacteria cell before we culture the bacteria. So we're using plasmids because they are not the main bit of DNA in the bacteria, so we can interfere with them and it has relatively limited impact on the bacteria. But when we're talking about the plasmid beforehand, we're actually talking about a plasmid that is outside of the bacteria cell, and then we have to insert it before we allow it to grow. We looked at the 3 methods of isolating a gene, the required gene we need. Today we're going to look at how. And in the last video we talked about restriction endonucleases, cutting the DNA, but cutting them at this staggered end, as you can see here, which gives this exposed bases that want to anneal to other exposed bases, which allow us to insert the DNA of the required gene into the plasmid. Now, obviously, this is a very low success rate, but it does help that we can use the same restriction endonuclease on the plasmid and the required gene to make complementary sticky ends, which will combine together. Once the sticky ends have been put in the same place, so this one, for example, with that one here, they will anneal naturally, but to completely seal this, we use the enzyme DNA ligase. How we do this? Well, first of all, we need to insert the DNA into an open plasmid. So we use the same restriction endonuclease, as I said a few times now, to leave sticky ends on the plasmid, which are complementary to the sticky ends on the required gene we're going to insert. But as well as the gene on either end of the gene, so here and here, we have a promoter region, which is where the RNA polymerase will need to attach when the protein is being made in protein synthesis, and a terminator region where the RNA polymerase will unattach in the same process. So we have these 2 extra sections either end of the DNA molecule that we're putting in. And obviously we stick the plasmid and the required gene in with DNA ligase for a process called ligation, and hopefully that will mean that the DNA and the required gene and the plasmid will seal together. Once we've done this, we need to insert the plasmid into the bacterial cell, and we use something called calcium chloride heat shock. So here, in order to do this, we put the bacteria in the plasmids, first of all, in ice cold calcium chloride. We then temporarily raise the temperature to 42 degrees for 2 minutes, which increases the permeability with the sudden change in temperature and with the charges of the calcium chloride. That will allow us then, hopefully, to insert the insert the plasmid into the bacteria cell. So by lowering it down to that ice cold temperature, adding its calcium chloride and increasing to 42 minutes, hopefully that plasmid will uptake into the bacteria. Always works. There are a couple of different potential outcomes, because only about 1% or less will actually take up the plasmid required when mixed. Some plasmids that are taken up will not actually have any required gene in it. They'll just closed up on themselves, so they've taken up a normal plasmid. and some of them will not have taken up any plasmids whatsoever, and obviously some of them will have taken up the recombinant plasmid to become transgenic bacteria. So we can test this using 3 techniques. The first technique we're going to look at in detail in a second, antibiotic resistant marker genes in a process called replica plating. The second technique is we could fluorescently mark the DNA in the gene before we do any of this, and therefore we'll be able to identify it. And the third technique is enzyme markers. So 1 and 3 I'm going to look at in detail with you. Fluorescent markers comes up when we look at DNA probes later in the videos. Replica plating them. So as you should know from earlier on in your school life, plasmids are used by bacteria naturally for antibiotic resistance. And 2 main examples that we use are used in a model that you need to know for this. So 2 main genes that are giving resistance, or 2 main antibiotics that need resistance, are ampicillin and tetracycline. So this is a regular plasmid, and this regular plasmid has 2 genes in it, one for ampicillin resistance, the blue one, and tetracycline resistance, the yellow one. So what we can do is we can work out, has a bacteria taken up a recombinant plasmid or any plasmid from what its resistance will be to different antibiotics. Because if the human insulin gene, and this is the example you need to know, is uptaken, the human insulin gene will insert itself in the middle of the tetracycline resistance gene due to the restriction endonuclease always cutting at the same point. So what this allows us to do is work out, has the required gene, in this case insulin, been uptaken in the plasmid, and then that's been uptaken by the bacteria. So how do we do this? Well, we grow colonies of bacteria, and then we transfer some of those colonies onto plates of agar containing the ampicillin or the tetracycline. Obviously, we still have the original colonies on a normal plate without these. So once we've identified them, we can go back and isolate the colonies that we want. So first of all, we grow them on an ampicillin agar. The ampicillin agar will mean that only bacteria that have uptaken a plasmid will be able to survive, because you need the plasmid and you need a plasmid with ampicillin resistance in it. Now, any bacteria taking up either type of plasmid will be resistant to ampicillin. So once we've done that, that eliminates any bacteria that have not taken up any plasmids. We then test the ones that have survived on the ampicillin and put them onto the tetracycline. So the ones that have survived the ampicillin but die on the tetracycline are the ones that have the human insulin gene inserted, because the ampicillin gene will work still, the tetracycline resistance gene is broken and therefore will not work. And then we can go back, isolate those colonies, and use them to identify which ones have the recombinant DNA. Similar method with enzymes. Now the details and the pictures here are not important, you only just need to be aware of that. And you can use the enzyme lactase, which breaks down lactose into glucose and galactose. And this this enzyme is found in many bacteria plasmids as well. And when we uptake certain genes, it can break the lactase gene in half, preventing it from being able to produce lactase, preventing then lactose being broken down into galactose. Bacterial colonies grown on agar with a particular type of gel called X-gel. And this gel is white initially, it contains lactose, but when it's digested into glucose and galactose because of the enzyme, it will go to a blue product. So ideally, the normal plasmid will break it down into the blue product, but if you've got a plasmid that has uptaken the required gene, it will not break down, and the recombinant plasmid will stay in the white colour. And whilst we don't study them in A-level because we focus on the bacterial method, you could have application questions linking to it. So you can see here bacteria has been used for everything from insulin to treating emphysema, to plants being used, to cattle that produce fibrogen in milk, to goats which produce antithrombin in the milk as well. So lots of uses of transgenic organisms now and potentially into the future that you could be given as examples in an exam.